Hi, hello, very happy good evening. So this is Krishna Ma'am and we've been talking about the story of life on earth, and that's biology. So here we are in the next highlight series. So henceforth our class timing will be at 5:30. So for all those who have been a little confused about my class timings, because it ranges from 3:15 to 4:30 then 5:30, so I'm really sorry about the change in timing. So it has to fit with the slots along with the other classes. So that is the reason there's a lot of confusion with the time slots. So for the next I would say for the next 3.5 weeks, so it will be only 5:30, so you don't have to worry much about. So we'll discuss all the brainstorming sessions here, all the tough concepts here. So here we are in the highlight series. So we are going to highlight the concept of ETC, that is electron transport chain. So this is one concept that is remaining as a part of your aerobic respiration. So we have been discussing aerobic respiration. The last part of your aerobic respiration is your ETC, that is electron transport chain. All right, yes, so shall we go? You ready? So I also hope you guys had a splendid weekend and you guys are seriously preparing for your exams and with a lot of commitment and spirit within. So I hope your preparations give you a lot of success. So let's move forward. So we're going to talk about ETC today. So it's very simple. So I'm not going to complicate with a lot of new terms. So we are not going to see anything extra with respect to ETC. So probably I would say glycolysis and Krebs cycle I have highlighted much in detail. But whereas ETC I will not detail it as much. So we'll stick to the level that is given in your NCERT. All right, yes, so here we go, ETC. So before we go to ETC, so you have to have a link between the previous thing and what we're going to talk today. So we have been we actually took off from this slide, if you remember, on Friday. So I started with the respiratory pathways on Friday and hence we took off further. So first of all, before the respiratory pathways happens in two different ways, in aerobic as well as anaerobic organism. But it begins with the primary process, that is your glycolysis. So glycolysis occurs in all living organisms, irrespective of whether they are living or they are non-living. All right. So after glycolysis, the fate divides into two, categorizing them as aerobic and as anaerobic organism. So if it's aerobic organism, after glycolysis, it undergoes a link reaction which links glycolysis and the next one, that is the Krebs cycle. So it is glycolysis, link reaction, Krebs cycle and your ETC for aerobic respiration. So we have already done with link reaction, Krebs cycle. So what is remaining is only electron transport chain. So if the fate decides it to be an anaerobic organism, so then we have fermentation. Right, so right after glycolysis they undergo fermentation, so producing either alcohol, as in case of yeast, which is termed as alcoholic fermentation, or as lactic acid, as in case of lactic acid bacteria or mussels. So this is how the entire process started and this is where we actually started it off with, right? So now we move further to give you to talk a quick review or recap about your Krebs cycle, which we discussed in the previous lecture. So here we go to a Krebs cycle. So I hope you don't get confused with the Krebs cycle. So it's pretty easy, it's not really tough. So here we talk about the Krebs cycle. Yeah, so it's a bit colorful, so we're having a lot of equations. But I hope after our lecture on Saturday, so Krebs cycle is still easy, it is no longer tough. Right? So here we go. So acetyl-CoA, so what happens in glycolysis? So in glycolysis, the respiratory substrate is glucose, right? So this glucose gets broken down into pyruvate or pyruvic acid. So one molecule of glucose breaks down to form two molecules of pyruvate or two molecules of pyruvic acid, right? Okay, so this pyruvic acid, so we're talking only with respect to the aerobic organism. So it enters what is called as a link reaction, wherein the pyruvate molecules are converted into two molecules of acetyl-CoA each. Right, and this happens in the presence of pyruvate dehydrogenase enzyme. It is a type of oxidative decarboxylation. So after that, your acetyl-CoA enters into this TCA cycle. So Manoj Singh, Manoj Singh, hello, hi. So thank you so much for joining my session today. So please do like, share and subscribe. So your S means that you have been listening to my previous lecture. So thank you for joining. All right. Yes, so this acetyl-CoA is now going to enter into Krebs cycle. So let me remind you, for one molecule of glucose, we get 2 molecules of pyruvate, and for each molecule of pyruvate, we have 1 round of Krebs cycle. So since we have 2 molecules of acetyl-CoA, so your Krebs cycle happens twice for 1 molecule of glucose. Okay, so yes, so this acetyl-CoA from the link reaction enters, and in the presence of citrate synthase, it forms your stable product, that is citrate. So citrate is a 6-carbon molecule, and this is a type of condensation reaction. So you know what is condensation. So here water is condensed to form the citrate. So acetyl-CoA comes out, and so this CoA portion comes out and it becomes citrate. So that is the first step. So the second step is that citrate gets converted into alpha-ketoglutaric acid. So from 6C it is becoming 5C. So this is to tell you there are 6 carbons. So this is to tell you there are 5 carbons. Alright. So citrate becomes alpha-ketoglutaric acid. Alright. So in the presence of isocitrate dehydrogenase, so it is this type of reaction is decarboxylation. Okay. You watched Hindi lectures, alright, so you find English a bit tough, that's completely fine. But if you want me to explain anything once again, so please do let me know, alright? Yeah, so this is a type of decarboxylation reaction. So what do I mean by decarboxylation reaction? Decarboxylation. So I remove 1 carbon or 1 carbon dioxide. So both are same, right? So 1 carbon means in the form of carbon dioxide. So this is 6C. So I remove 1 carbon dioxide. So it becomes 5C. So this becomes alpha-ketoglutaric acid, and I get NADH plus H plus as a result. I am not utilizing it. Okay. So after that, the alpha-ketoglutaric acid gets converted into succinyl-CoA. This is an intermediate step. So right afterwards, succinyl-CoA is converted into succinate. So succinate is a 4-carbon compound. So this was a 5-carbon compound. So this is a 4-carbon compound. So this intermediate step is catalyzed by dehydrogenase complex, alpha-ketoglutaric dehydrogenase complex. So it's again a type of decarboxylation reaction. So what is oxidative decarboxylation? So when NADH is getting converted into NADH plus H plus, this is oxidation. And when 1 carbon is removed, so that is decarboxylation. So this entire thing is oxidative decarboxylation. You got the trick? Yes. So here also it says decarboxylation, decarboxylation. I hope you now know what is decarboxylation. So when I say oxidative decarboxylation, iska matlab hai when NAD plus is getting converted to NADH plus H plus. Okay. Alright, so now your succinyl-CoA getting converted into succinate in the presence of succinyl-CoA synthetase is a type of substrate level phosphorylation. So substrate level phosphorylation is the step or is the reaction where your ATP is gained. So irrespective of whether you have gold, you have diamond, you have cash, always if you want to buy something, we buy in terms of cash. So I cannot say I have diamonds, so you give me this. So even if you have diamonds, it has to be converted into cash, the currency that is the need of the hour, and then you get your goods, right? So much in the same way, whichever step gives you the ATP, the energy currency of the body, so that is your substrate level phosphorylation. So that is in this step. So in your textbook, you don't have these two, right? So you don't have the succinyl-CoA step. So in your textbook, it directly says alpha-ketoglutaric acid to succinate, but it's okay if you have to remember one intermediate step. So the main thing that if you have to remember, if you have to understand this, you have to remember what type of reaction all these are. So condensation, decarboxylation, oxidative decarboxylation, and substrate level phosphorylation. No ma'am, I watched your Hindi lecture. Okay, you feel my English, okay, your English is becoming worse day by day. Okay, so you watched my Hindi lecture, but I don't teach in Hindi. So where did you watch my Hindi lecture? Alright, anyways, cool. Okay, so finally we are here in succinate, which is a 4-carbon compound. So by now we have 2 NADPH and 1 GTP. So GTP is guanine triphosphate, which is equivalent to your ATP, that is adenosine triphosphate. So both are nucleotides, so it does not matter if it is GTP or your ATP. Fine. So I am moving more further. So succinate becomes fumarate in the next step. So fumarate is again 4-carbon. Yes. Okay. So this is in the presence of succinate dehydrogenase. So this is the type of dehydrogenation reaction. So okay, so you watched my lecture in Hindi last year. Oh my god, I don't know when I spoke in Hindi. Alright. So this is a type of dehydrogenation reaction wherein you remove one hydrogen on this side and put it on the other side, because ultimately both are 4C carbons, right? So here you gain FADH plus H plus. Okay, so then your fumarate gets converted into malate. Again, it's a type of hydration reaction. So in the presence of fumarase. Then finally your malate gets converted into oxaloacetic acid, which is your 4-carbon compound. In the presence of malate dehydrogenase, this is dehydrogenation. So whenever it is decarboxylation, you have removal of 1 carbon. Whenever it is dehydrogenation, wherein you remove a H molecule from the substrate. So here again you have the gain of NADH. So if you see for 1 molecule of acetyl-CoA, we have 3 NADH. 2 was in the previous slide, 1 here, 1 FADH, 1 ATP. If you remember, we elucidated it in the last class. So this is how your TCA is. Okay. Okay, what is FADPH and what is FAD? Alright, so when there is FADPH, flavin adenine phosphate, so that means it is used for catabolic reactions. When it is FADH, it is used for anabolic reactions. So there's always a difference. So if you see NAD also, it is NADPH or NADH. So what is this, ma'am? Should we write P or should we write, we should not write P? Is it only NADH? Yes, this question comes a lot of time. That is because when there is P, it is used in catabolic reaction. When there is no P, it is used in anabolic reaction. So I hope you know the difference between catabolic and anabolic reaction. So catabolic is breaking down bigger molecules to form smaller ones. Anabolic is smaller molecules building up to form a larger molecule. Alright, Manu? Yes, thank you so much for joining my session. So please do like, share and subscribe. Okay, so for 1 molecule of acetyl-CoA, so we have 3 molecules of NADH plus H plus you have 1 molecule of FADH plus H plus. So this is 3H plus and 1 molecule of GTP, which is equivalent to ATP. But at the end of link reaction, you have 2 molecules of acetyl-CoA. So which means this cycle happens twice, right? So this is for 1 cycle of 1 round of Krebs cycle. Yes, ma'am, a very happy good evening. So thank you so much for joining my session. So please do like, share and subscribe. Okay, so now we are writing for 2 molecules of acetyl-CoA. So here it will be into 2. So do everything into 2. So it will be 6 molecules of NADH plus 6 molecules of H plus. So then you have 2 molecules of FADH. So plus 2 molecules of ATP. So this is the net energy gain as a part of your Krebs cycle. Okay, so what are the end products of Krebs cycle? You have oxaloacetic acid plus carbon dioxide coming out. So remember, for your photosynthesis, the equation is carbon dioxide plus water, which forms your glucose, right? So this is C6H12O6 plus you have oxygen. So smaller molecules are forming larger molecules. So this is a type of anabolic reaction. So here you have only NADP, right? In respiration, larger molecules are broken down to form smaller molecules. So it is NADPH, okay, right? So this is glucose. So this is the equation of photosynthesis. So what happens in respiration? So your C6H12, it breaks down to give you water plus carbon dioxide plus some amount of energy. So this carbon dioxide comes out during the Krebs cycle. Okay, so the water comes out during the electron transport system. So this is what is happening. So at the end of Krebs cycle, so you get your oxaloacetic acid plus carbon dioxide. So your end product carbon dioxide is obtained. Yes. Hi Rose Amu, I guess. So hello Rose, yes, a very happy good evening. So thank you so much for joining my session today. So please don't forget to like, share and subscribe. Okay, fine. Okay, so this is at the end of your Krebs cycle. So okay, so I've come to ETS. So quickly I'll give you a summary of how much electrons, how much ATP is gained in glycolysis. So quickly we'll see it and then we'll move to ETS. So at the end of glycolysis, so we have 2 ATP plus we have 2 NADH. So I am talking only about the gain. Okay. So then we have the link reaction. So here we are talking for 1 molecule of glucose. So this gives 2 molecules of pyruvic acid. So here 2 molecules of pyruvic acid gives 2 molecules of acetyl-CoA. Right? So here it is 2 NADH plus 2H plus. For 2 molecules it is 2 NADH, right? So now talking about the next one, that is your Krebs cycle. So for Krebs cycle, you have 2 rounds of Krebs cycle because you have 2 molecules of acetyl-CoA. So it would be what? So it will be 6 NADH plus 6H plus. So it is 2 FADH plus 2H plus you have 2 ATP. So this is for aerobic respiration and this is also for 1 molecule of glucose. Yes? Okay. So now understand. So we can convert NADH also in terms of your ATP. So 1 NADH is equal to 3 molecules of ATP. So 1 FADH is equal to 2 molecules of ATP. Fine? So now convert. So first of all, let's count how many NADH we have and then we'll convert them. So I have 2 NADH, 2, 4. 4 plus 6, so totally we have 10 NADH. So let's leave off H plus as of now because that is not going to give us energy. Okay, so now let's count the ATP. So 2 ATP, 2 ATP. So it's going to be plus 4 ATP. So how many FADH we have? We have only 2 FADH. So now convert everything in terms of ATP. So 1 NADH is equal to 3 ATP. So into 3, so this will be 30 ATP. So this is in terms of ATP. So I write as such. Now FAD, so into 2. So how much I have? I have 4 FAD. So it will be 4 ATP, sorry. So now count how many ATP we have? 34 plus 4. So totally at the end of aerobic respiration, before your ETC, you have 38 ATP molecules. Clear? Yes? Any doubt? So far you're clear with aerobic respiration. Can we go to the last step in aerobic respiration, that is your ETS, that is electron transport system. So you understood how many molecules of ATP we are getting at the end of aerobic respiration. So is this entire calculation clear? Yes. So now we move forward to talk about your ETC molecule. So what is ETS? That is Electron Transport System or Electron Transport Chain. So people say it in two ways, either as ETS, Electron Transport System, just like you say your BTS, or ETC, etc. So that is Electron Transport Chain. So chain and system is what varies. You can say it either way, it's not wrong. Okay, so why it has to happen? To release and utilize the energy stored in NADH plus H plus and FADH plus H plus. So I told you, no, irrespective of whether you have gold and diamond, though gold and diamond value more than the normal cash, ultimately if you want to buy anything, you can buy only in terms of cash. Even if you have gold, you have to convert it into cash and buy it. Right? So similarly, your body can only utilize the energy stored in the form of ATP. Though NADH and FADH contribute to higher energy molecules, it has to be converted into ATP. So now here in the step of ETS, we are going to convert the energy that is trapped inside NADH and FADH. Alright? Yes? So let's see how. So it is accomplished by oxidation through the electron transport system. So now how are we going to convert the energy that is trapped inside NADH or FADH is to oxidize them through the ETC. So we are going to oxidize them. So electrons are passed on to the oxygen, resulting in the formation of water. So when I wrote the equation of respiration, so the end products were water and carbon dioxide. So carbon dioxide was obtained in the Krebs cycle. Now water we have to obtain. So that final water molecule is produced in the ETS, and how? Through oxygen. Right? Yes, the metabolic pathway through which the electron passes from one carrier to the other. So there are so many complexes, so the electron passes one after the other. So this is why it is called as electron transport system. So electrons are transported through a series of complexes one after the other. So that is why it is called as the electron transport system. Alright, clear? Okay, so let's move forward. So this ETS is present in the inner mitochondrial membrane. So giving you a quick recap. So glycolysis happens where? So glycolysis happens in the cytoplasm. So where does your link reaction happen? In the mitochondrial matrix, that is the cytoplasm of the mitochondria. So where does Krebs cycle happen? In the same location. Now where is ETS happening? Inner mitochondrial membrane. So we have mitochondria as a double membrane structure. So we have an outer membrane and we have an inner membrane, right? So this space is the intermembrane space. So your ETS is present on this inner membrane. Okay, fine, right. So we move forward. So your electrons moves through a series of complexes. So that is why it is called as the electron transport system. So if you see, this is the cytosol, meaning the cytoplasm. The cytoplasm, so you can say this is on the outer side. Okay, so this is the space, intermembrane space. So the matrix is the inner portion of the mitochondria. So in the matrix you had your Krebs cycle happening, you have your link reaction happening. So here you have your inner mitochondrial membrane, and this is where the complexes are embedded, right? So outside this you have the intermembrane space, but they have written cytosol, don't get confused, right? So your electrons move from complex 1 to complex 3, complex 2 to 3, then it moves to complex 4 and finally complex 5. So this is how it moves, and finally we get water as well as you get ATP. That is the entire concept of your ETS. Fine? Okay, so let's move forward. So who are the complexes of your ETS? There are 4 main complexes. The last one I'll explain towards the last slide. Okay. So there are 4 main complexes for your ETC. So what are those complexes? First is complex 1. So complex 1 is named as the NADH dehydrogenase complex. It is called as the NADH dehydrogenase complex. So I have taught you what is dehydrogenase. So it is this removal of hydrogen, right? Dehydrogenase is as simple as that. So complex 2 is succinate dehydrogenase. So if you remember Saturday's class, I told you when we started Krebs cycle that all the enzymes for Krebs cycle are present in the matrix of the mitochondria except one. So that one is your succinate dehydrogenase, which is present on the inner mitochondrial membrane. So complex 2 is called as your succinate dehydrogenase. Fine. So complex 3 is called as the cytochrome BC1 complex. So electron transports, you have cytochrome, you have seen it in your Z scheme where you have PS1 and PS2. There also you have electrons, that is how they move through, right? So you know cytochrome is a component of iron sulfur clusters, right? So you have cytochrome B and C as complex 3. So now what is the last complex? That is your cytochrome C oxidase complex. So these are the 4 main complex, and the last one which is not mentioned here is your ATPase. So I hope you know ATPase. It's a part of chemiosmotic hypothesis that we read. So in chemiosmotic hypothesis we saw how ATP is made, right? So to make ATP we have to have a proton gradient. So similarly we are going to have a proton gradient here, but how are we going to create the proton gradient? Is through oxidation reduction, okay, mostly through oxidation. So that is why it is also called as oxidative phosphorylation. Fine, cool. So now we go. So this is how it is. So if you see, this is your complex 3. So from complex 1 as well as from complex 2, the electrons move towards complex 3, which is your cytochrome BC complex. From this cytochrome BC complex, it moves into complex 4 and then finally into the complex 5, which is your ATP synthase. So we'll come here, hold on. So if you see, we are trying to utilize the energy that is stored in NADH, right? So this NADH is produced in the matrix as a part of your Krebs cycle and your link reaction. So we have totally 10 NADH, right? So this NADH is now getting reduced to NAD+, right? So NAD is getting oxidized. So this is like this. So when I do reverse, so this is oxidation, right? Removal of hydrogen is oxidation, right? So now your NADH becomes NAD+. Plus you have 2H plus coming out. So along with two electrons also coming out. So this H plus and this electrons moves inside this complex. So these 2 electrons now combine with FMN. Okay, so this FMN now combines with FES. So when it combines with FES, it releases 2 more electrons and 2 more protons. So totally I have 2H plus here and 2H plus here. Both the H plus comes to the inner membrane space. So totally I have 4H plus. So pay attention, NADH is getting converted into NAD. Plus I have 2H plus, this H and this H, along with the release of electrons. So this electrons now combines with FMN. When it combines with FMN, later with FES, it again releases 2 more electrons and H plus. The electrons will move through the carriers, but the 4 H plus that is remaining will come into the intermembrane space. Fine, so this is how it starts, but let's look into the next slide. So we'll read the theory, so it will be easy line by line. The electrons present in the NADH, which is in the mitochondrial matrix, produced during the citric acid, it is oxidized by NADH dehydrogenase. So it is oxidized, right? So removal of hydrogen. So NADH plus H plus is getting oxidized into NAD plus 2H plus. So that is why this is oxidative phosphorylation. So the electrons are transferred into a carrier. So the complexes are situated or embedded in the inner mitochondrial membrane. So if I am one building, you are another building, if you have to travel from one building to the other building, you take a cab or you walk, right? So similarly, how will electrons move from one complex to the other complex is with the help of electron carriers once again. One such carrier is your ubiquinone. So ubiquinone is a carrier. So electrons are transferred through ubiquinone located with the inner membrane. If you see, sometimes two buildings are connected together with an interbridge. So imagine ubiquinone as an interbridge, alright? Yes, so now the electrons from this oxidation goes into ubiquinone. Fine. So now I am moving further. Ubiquinone receives same electron equivalence when FADH breaks down. So complex 1 NADH is oxidized, complex 2 FADH is oxidized. So both will release H plus as well as electrons. The electrons are collected by the ubiquinone. So your FADH is oxidized in the presence of an enzyme succinate dehydrogenase, whereas this is oxidized in the presence of NADH dehydrogenase, meaning we are removing hydrogen. Fine. So reduced ubiquinone. So when lot of electrons enter ubiquinone, it is not really active. It enters the reduced state, that is ubiquinol. So now this ubiquinol is oxidized. So it transfers electrons to the cytochrome C via the cytochrome Bc1 complex. Now when it has more electrons, all that it has to do is that it has to push it to the next carrier. The next carrier is your cytochrome C actually cytochrome Bc1, and then to your cytochrome oxidase. So the electron keeps on moving, right? So the cytochrome C is a small protein attached to the outer surface of the inner membrane. So cytochrome oxidase, right? It acts as a mobile carrier for the transfer of electrons between complex 3 and complex 4. Between complex 1 to complex 3, between complex 2 to complex 3, we have ubiquinone. This is your first carrier. The second carrier is the cytochrome C. So you have complex 1, you have complex 2, 3 and 4. So from here to here and from here to here you have ubiquinone. From here to here you have this cytochrome C carrier. So that is all that it makes sense here. Fine. Is it not simple? So we go to the next path. So complex 4, that is your cytochrome C oxidase, is a complex containing cytochrome A and A3 and copper centers. So this is talking about the components of complex 4. So when the electrons pass from one carrier to the other carrier via complex 1 to 4, the electrons are finally coupled to the ATP synthase. So after it reaches all the 4 complexes, it reaches the 5th one, that is your ATPase synthase. So this ATPase synthase, we have read it in various ways. One in the head of myoglobin, next in the chemiosmotic hypothesis. ATP synthase, it can synthesize ATP, or it can actually take up ATP and break it down. So here it is going to make ATP from ADP plus Pi. Alright, yes. So number of ATP molecules depend on the nature of the electron donor. The role of oxygen is limited to the terminal stage of the process. So where does this oxygen come from? From photosynthesis. I hope you are able to correlate. Yes, oxygen acts as the final hydrogen acceptor. So as you see, as a part of NAD and FAD oxidation, H plus is getting accumulated in your intermembrane space. So you have more amount of H plus in the intermembrane space than in the matrix. So a gradient is formed. Yes. Now who is going to accept this hydrogen to make ATP is your oxygen. Is it not beautiful? So these are naturally created processes which you and I, when thought otherwise, would not make it happen, right? So energy of oxidation reduction is utilized in this proton gradient. So that is why it is called as oxidative phosphorylation. Okay, fine. So we'll come to this. Right. So energy released when the electron is utilized in synthesizing ATP. So now energy is released when the electrons are transported, right? So the last complex. So you have coupled the movement of electrons to energy synthesis by creating a proton gradient. So now this proton gradient has to break in the presence of complex 5. So complex 5 has two components, F1 and F0. So F1 is the peripheral protein complex. It contains the site for ATP synthase. F0 is an integral membrane protein that forms a complex channel. So once again we go to the previous slide. See, so this is the last complex, that is complex 5, which is ATP synthase. So you have F0, you have F1. So this is on the outer side, so this is peripheral membrane protein. This can synthesize ATP. If you see, when it gets ADP plus PI, it can synthesize ATP. Whereas F0 is an integral membrane complex because it is embedded on the membrane, right? Okay, so now we go further. Okay, for each ATP produced, 4H plus passes through the F0. So 4H plus ions pass through the F0, which is an integral part. So F0 is here, F1 is here. So when 4H plus passes like this, so the conformational change happens, because of which ADP plus PI is converted into ATP. Fine. So that is what is mentioned here. Is ETC still tough if you understand the logic behind it? Yes. Is it not easy now? So I hope you have understood. So you have complex 1, you have complex 2, complex 3, 4 and 5. So who is the carrier from here to here and here to here? Ubiquinone. So when this is loaded with electrons, it becomes ubiquinol. Who is the carrier here? Cytochrome C. Sorry, So who is the carrier here? So this is cytochrome C. And finally, this has two parts, F0 and F1. Right? So we are done with your ETS. So in a nutshell, we are done with your respiratory pathways. So I have given you highlights on photosynthetic pathways C2, C3, C4 and CAM plants. And then I moved on to respiratory pathways, where I spoke to you about glycolysis, Krebs cycle, link reaction, your fermentation, your ETS. So tomorrow we have a session talking about digestive ducts and the enzymes used in digestion. So tomorrow again the class is at 5:30 p.m. So please join me. So we are going to have a quick recap of the ducts in digestion as well as the enzymes that is utilized in digestion. Alright. So I hope now your ETC is no longer tough. It is pretty easy. Yes. So if you genuinely like my session, so please do like, share and subscribe. And thank you so much for watching my session. So I'll be here tomorrow at 5:30. So until then, stay tuned and continue your preparations. So thank you and take care.